I would now like to welcome to the floor Professor Gordan Lauc, Chief Science Officer and co-founder of GlycanAge, who will be speaking on the role of glycans as biomarkers of aging and their role in preventing inflammation in particular. Professor Lauc, thank you so much for joining. Pleasure to be hosting you today. The floor is yours.
Thank you, Franco. I will tell you something about glycans as the biomarkers, but even more importantly, as the functional effectors of aging. And ask one of the key questions is, can we make our glycans young again? And can we in this way prevent inflammation and keep people healthy for the most of their lives? So glycans are the ultimate layer of molecular complexity. Unfortunately, in the same time, they're the most neglected molecules of cellular communication, and this became very evident in this pandemics when even the director of NIH was writing about as glycoprotein without a single glycan on its surface, while actually this protein is more glycan than peptide on the surface. And these glycans are not only decorations, they're actually actively participating in the interaction between the S-glycoprotein and its receptors. And this is not only for the S-glycoprotein, this is for the majority of proteins that we have in and on our cells. And to see the complete picture, to understand biology, we have to include glycans in what we are doing. And when we think about glycans, they're part of a protein. So adding a different glycan in a process called alternative glycosylation is functionally analogous to coding mutations. So whether we know that mutation can kill a person, but also can alternative glycosylation. The key difference is glycans are inherited as a complex trait. So there are dozens of genes which form a network which decides about the specific glycosylation. And this is a network which codes for IgG glycosylation, as we have discovered in several of our papers. So glycans integrate genetic information, epigenetic information, and direct environmental information into a molecular structure. So if glycans are so cool, if they integrate so much information, why are they being so neglected? Why are no more people working in the field of glycomics? And the reason is that this is difficult. Glycan analysis is very complex. Glycans are nonlinear molecules, and the technology which enables analysis of a large number of samples is available for only a little bit more than a decade. And my lab is leading this field, and we have already analyzed glycans in over 150,000 samples, but not any samples. We did it in collaboration with some of the leading researchers from the leading institutions all around the world, and generated a huge amount of data, which we are now using in multiple areas, including the longevity field. In the field of longevity, we focus on a specific glycans attached to immunoglobulin G, where we know that they have multiple roles in balancing inflammation, both directly affecting the effector functions of IgG. So where we have glycans which are acting as anti-inflammatory, suppressing inflammation, while other glycans will make the same IgG very pro-inflammatory, but also in a multiple other levels where glycans interact with cells and affect cell recognition and so on. And based on these immunoglobulin glycans, we developed something we call the GlycanAge, which is a test that integrates genetic, epigenetic, and environmental factors in a simple index that can be reliably quantified in a high throughput manner. And as David Sinclair and I published in an editorial in Aging last year, this is molecules which are functional effectors of inflammation. They either promote or suppress low-grade chronic inflammation. So we know that if we have these young glycans, they're actively suppressing inflammation, while we have these old glycans, they're actually promoting inflammation. And this is now formed in a form of a commercial available GlycanAge test. We have a startup which is commercializing this globally. It's a London-based company. It's still a small startup, but it is backed by the R&D in my main company, Genos, where you have 50 plus researchers working in the field. So what we have learned so far is that changes in IgG glycan in many diseases resemble aging. So the last 4 columns are aging. So this is what happens with glycans as we are getting old. And all other columns are different diseases. So people with different diseases have a similar changes in IgG glycan. And in many aspects, these changes come much before the disease. So in some examples, we see changes in the glycans up to a decade before the disease actually develops. And glycans, by their role in the regulation of low-grade chronic inflammation, are functional effectors which actually contribute to the disease. And this is all backed by a lot of science. We have published over 150 papers so far, some of them very prominent, including the circulation paper, several gastroenterology papers, several Nature group papers. So all this has a long and strong scientific backing. And of course, this was enabled by a very generous funding from different funding agencies. We got over 15 million euros in research grants, which enabled us to perform all this analysis. And now the key question, how can we revert it? So what can we do to change the glycan age clock to go backwards? And in this way, stop inflammaging, stop low-grade chronic inflammation, and keep people healthy. So we know that things are doable, and probably the most successful person in anti-aging regarding the glycan age is Joseph Raphael and his PhysioAge clinic in New York, where we have already analyzed over 150 of his patients, and most of his patients are 20 to 40 years younger in the glycan age. And often we see something like that somebody comes with relatively bad glycan age, and after a couple of months of his therapy, their glycan age heavily improves. And of course, the key question is this relevant? Is this important? So if you have a better glycan age, does it really make you healthier? So we are doing research to confirm that. And one of the strongest evidence we have so far is a circulation paper we published slightly less than 2 years ago, where we have shown that these bad IgG glycans are actually causative for development of hypertension. And if you fix them, and they can be fixed in mice by feeding N-acetylmanosamine, even the obese mice would not develop hypertension. This was in mice. In humans, we have shown the same correlations. We still do not have the proof of causation, but we just got a paper accepted. It's not out yet. It will be out in a few weeks, which show that IgG glycans are actually predictive of hypertension even in humans. We also did an interesting prospective cohort where the samples were collected over 30 years ago and then follow up longitudinally. And in that period, there were 508 cases of either heart attack or stroke. And when we looked at IgG glycans, they were as predictive as the entire AHA score. And in women, even a single glycan, this structure here, was more predictive than the AHA score. So actually, glycans do predict future cardiovascular events, and probably through regulation of low-grade chronic inflammation, they actually also participate in development of cardiovascular diseases. These things can be improved in various ways. Something which has been shown to be very effective is the weight loss. And for example, in bariatric surgery, people who lose 20, 30 kilos after surgery, we had a patient who improved for 37 glycan years in just 6 months. Not everybody improved that much, but in general, people improve after bariatric surgery, but also they improve if they lose weight normally. We were tracking 2,000 twins in 3 time points over 15 years together with Tim Spector. And we have shown that people who were gaining weight were aging faster than people who were losing weight who were aging slowlier. Unfortunately, there is no magic diet. We had a study of 1,000 people on 5 different diets, and each of the diets was beneficial for some people, and it was not beneficial for other people. So everybody is different, and there is no magic solution. So we have to try, and we can use different biomarkers, including, of course, the glycan age, to see what works. Physical activity can also help, but can also be detrimental. So too much exercise, overtraining usually results in more inflammation and worse glycan age. Something what is extremely efficient in women is estrogen. We had properly designed randomized placebo-controlled trial where the menopause was chemically induced. And then the supplementation of estrogen prevented any effect, and the woman given placebo aged on average 9 years within 6 months, which then recovered after the recovery of the gonadal hormones. And interestingly, the woman who had the best glycan age at the beginning of the study changed the most, while those who were worse already did not change that much. So estrogen is definitely one of the strong effectors. In the interest of time, I will just point to the recent review, which we published in the Comprehensive Glycoscience. And here you can see all the details about our last work, and it's freely available at this address here. And the key point, which I think we all want to make here, is that yes, with age comes diseases, different type of chronic diseases. And unfortunately, in our current healthcare system, people go to hospital when they are ill, when something does not function, and this is usually non-fixable. So this is why we call them chronic diseases. But if we detect the problem here, where only the biomarkers started to change, for example, when immunoglobulin glycans start to be pro-inflammatory, if we can fix them either with a lifestyle or some kind of pharmacological interventions, then maybe we will never end up with the disease and will continue to be healthy for decades to come. And glycan age can definitely help here, because one of the key problems why people do not do difficult lifestyle decisions is that the reward comes so late. So I have to do something today and be rewarded in several decades, and this can always be postponed for tomorrow. And with a test which can give feedback much faster, we can see that our hard work today actually pays back, and the glycan age can help here because it gives objective insight into the current state of low-grade chronic inflammation, and it's a quantitative tool which can help evaluate effects of lifestyle interventions. As I mentioned, it's commercially available. It can be purchased on our website. Sample collection kit comes by mail. It's a simple dried blood stain which can be mailed back, and then the samples are analyzed and returned into your portal. And I thank you for your attention, and I look forward to our discussion at the end of all talks.
Thank you so much for that, Professor Lauc. That was very insightful. I see a theme and a trend emerging with regards to blood-based biomarkers and biomarkers that are obtainable from blood. So that's a happy coincidence, as it were.